Today we're going to cover meiosis and sexual life cycles. Before the lecture begins, make sure you've read the assigned materials and taken notes, as well as prepared an outline for this lecture. During the lecture, minimize distractions by turning off any extra devices or background noise. Also, make sure that during the lecture you take plenty of notes, putting the slides in your own words in order to maximize your learning. First, we're going to get started by covering the inheritance of genes. Living organisms reproduce their own kind. When we talk about heredity, we are referring to the passing down or the transmission of traits, a term from Mendel, from one generation to the next. There are often differences from the parent generation to the offspring, and differences among the offspring as well. These differences are referred to as variation. The study of heredity and variation is known as genetics. Recall that Mendel studied the varying traits of the pea plants. These traits later became known as genes. They are the units of heredity. Genes are made up of segments of DNA which is stored in the nucleus. Genes are passed from one generation to the next. In order for genes to pass on, they undergo one of three forms of reproduction: asexual reproduction, cloning, or sexual reproduction. Asexual reproduction is when a single individual passes genes to the offspring. A clone is an offspring that is genetically identical to the parent. And in sexual reproduction, two gametes, one sperm and one egg, join together to give rise to the offspring. Humans have two types of cells: somatic cells and gamete cells. Gametes are the eggs and sperm. Somatic cells are the non-reproductive cells of the body, everything except the eggs and the sperm. Human somatic cells have 23 pairs of chromosomes. These pairs of chromosomes are known as sister chromatids. They are also called homologous chromosomes or homologs. Chromosomes in a homologous pair are the same length, the same shape, and the homologous pair carry the same genes and DNA. Homologous chromosomes are held together at the center by a centromere. So a total of 46 chromosomes in all somatic cells exist, but there are 23 pairs. Scientists can study these chromosomes through a karyotype, which is shown at the bottom right. All cells contain sex chromosomes, which determine the sex of the individual. Human females have a pair of homologous chromosomes, two X's. Human males also have a pair, one X and one Y. In somatic cells, 22 of the chromosomes are autosomes, or non-sex chromosomes, and one of the pair of chromosomes are the sex chromosomes, either the XX or the XY, which determines the sex of the individual. Each pair of homologous chromosomes include one chromosome from each parent. So in the karyotype below, imagine that the chromosomes on the left of each pair of homologous chromosomes is from the father, and the chromosome on the right of each pair is from the mother. Somatic cells are diploid, which is written out as 2N. It means that somatic cells have two sets of chromosomes. For humans, the diploid number is 46. One set of chromosomes would be 23. We get 23 chromosomes from each parent, and our somatic cells keep both copies so that they have two sets in each somatic cell. Somatic cells, therefore, are diploid. We'll discuss this more in a minute, but gamete cells are not diploid. Gamete cells, the egg and the sperm, are haploid. They contain 23 chromosomes, not 23 pairs like the somatic cells, but gametes contain 23 chromosomes. Now we're going to talk about sexual life cycles. A biological life cycle is the generation-to-generation sequence of events in the reproductive history of an organism. What are some of these events? Fertilization is when the egg and the sperm, the two gamete cells, fuse together to give rise to an offspring. The fertilized egg is known as a zygote and has a set of chromosomes from each parent. The zygote uses mitosis to generate the cells necessary for human development. When the human reaches sexual maturity, they produce gamete cells, which are haploid, containing only one set of chromosomes. Gametes are the only cells in humans that are produced through the process of meiosis, not mitosis. Meiosis results in only one set of chromosomes in each gamete, so each gamete has a haploid number. For all organisms that reproduce sexually, the alteration of meiosis and fertilization is common. However, there are some differences in the timing of these events. The figure to the lower left shows the timing of meiosis and fertilization in animals. In animals, gametes are the only haploid cell, the only cells containing 23 chromosomes. Gametes are produced by meiosis and do not divide before fertilization. Gametes, the egg and the sperm, both of which are haploid cells, fuse and form a diploid zygote, a zygote with 23 chromosomes from each gamete. Then, through mitosis, animals develop into a multicellular organism. For all organisms that reproduce sexually, the alteration of meiosis and fertilization is common. However, there are some differences in the timing of these events. The figure on the left shows the timeline for plants and some algae. Plants and a few algae have a life cycle that includes both a diploid and a haploid stage. The diploid organism is known as a sporophyte, which makes haploid spores through meiosis. After that, each spore grows by mitosis into a haploid organism known as a gametophyte. And then the gametophyte makes a haploid gamete through mitosis. Then fertilization of the gametes results in a diploid sporophyte. For all organisms that reproduce sexually, the alteration of meiosis and fertilization is common. However, there are differences when we look at the individual timing. If we look at the reproduction of fungi and protists, in most fungi and in some protists, there is a diploid stage that is a single-cell zygote, which produces haploid cells through meiosis. Each haploid cell grows by mitosis into a haploid multicellular organism. The haploid adult produces gametes through mitosis. Now let's look at meiosis. By way of review, once a cell is ready to divide, the chromatin condenses into chromosomes. The micrograph here shows a eukaryotic cell's chromosome. They are stained in purple. The micrograph on this slide shows a representation of the chromosomes that have undergone this condensation process. Once duplicated, each chromosome has two sister chromatids that are joined together. The structure looks like an X. The centromere is where the two sister chromatids connect. Again, the genetic material must make a copy of itself and condense to form chromosomes. Once duplicated, each chromosome has two sister chromatids. These chromatids are connected by a centromere, and the sister chromatids look like an X. Meiosis is similar to mitosis in that there is a duplication of the chromosomes. Meiosis takes place in two sets of divisions, meiosis I and meiosis II. The two divisions result in 4 daughter cells, unlike mitosis. Each daughter cell at the end of meiosis is haploid. It only has half as many chromosomes as the diploid parent. If we examine meiosis with only one pair of homologous chromosomes, as shown in the figure on the right, both individual pairs are duplicated. The resulting sister chromatids are very closely associated. The homologs may have different versions of each gene. A gene version is known as an allele. So in interphase, in meiosis, a pair of homologous chromosomes is duplicated. During meiosis 1, the homologous chromosomes are duplicated and undergo a division that results in haploid cells with duplicated chromosomes. And then in meiosis 2, the sister chromatids are separated, which result in haploid cells with unduplicated chromosomes. The process of both divisions of meiosis halves the number of chromosomes to make the haploid gamete cells. Meiosis reduces the number of each set of chromosomes from 2 to 1. This is so when the egg and the sperm combine, there are chromosome pairs. Without this reduction from a diploid to a haploid, we would have too many chromosomes. So for meiosis 1, the homologous pair of chromosomes pair up, and then they separate. Then, in the second division, the sister chromatids for each chromosome separate, resulting in 4 new haploid cells. This is an overview of the 2 divisions of meiosis. Remember, meiosis 1 separates the homologous chromosomes, and meiosis 2 separates the sister chromatids. That is the main distinction between meiosis 1 and meiosis 2. You could look under a microscope and be able to determine which stage of meiosis you were in, meiosis 1 or meiosis 2, based on the chromosome location and the chromosome number. To prepare yourself for the quiz and the test on this material, you will want to look at these cells out of order and without labels and determine whether or not you can identify which stage of meiosis the cell is in. In this figure, we will look at an overview of each of the steps of meiosis 1. One cell is entering meiosis 1 with 3 pairs of sister chromatids that have duplicated and made a pair of homologous chromosomes. You can see the red and the blue chromosomes. In prophase, the nucleus breaks down. In metaphase 1, the pairs of homologous chromosomes line up at the metaphase plate. They do so by the kinetochores interacting with the centromere in the center of the homologous pairs. The kinetochore microtubules are attached to the centrosomes at the poles. In anaphase 1, the pairs are separated to the poles where the centrosomes are by trafficking up the kinetochore microtubules. And then 2 cells are formed in telophase 1 and cytokinesis with a single chromosome pair. So looking at each step, let's consider prophase 1. The chromosomes are condensed and the homologous chromosome pairs align gene by gene. In meiosis, there are crossing over events. This can occur when non-sister chromatids, in the figure above, the blue is one set of sister chromatids and the red is another set of sister chromatids. So when these non-sister chromatids exchange DNA segments, this is known as crossing over. In the figure above, if the blue sister chromatid crossed over with the red sister chromatid, you would have a crossing over event. Each pair of homologous pairs has one or more X-shaped regions called a chiasmata. And if at the chiasmata, it's at this chiasmata where the crossing over between the non-sister chromatids occurs. Looking further at crossing over in prophase, this event produces recombinant chromosomes. This means a given chromosome that originated from one of the parents contains chromosomal information from both parents. In humans, approximately 1 to 3 crossing over events occur per chromosome. Crossing over contributes to the genetic variation seen in the human population, ensuring that the offspring are not simply clones of one parent or the other. Moving on from Prophase 1 into Metaphase 1. In Metaphase 1, the homologous pairs of sister chromatids line up at the metaphase plate in the middle of the cell, with a set of sister chromatids facing one cell and another set facing the other pole of the cell. Kinetochore microtubules are attached to the sister chromatids at the kinetochore. In Anaphase 1, the homologous pairs are separated. One sister chromatid moves towards a pole and the other moves towards the opposite pole. This movement on the kinetochore microtubules and poles are defined by the centromeres. In Telophase 1 and in cytokinesis, the cell begins to divide in half. Each chromosome still consists of the sister chromatids, and cytokinesis occurs, forming 2 haploid daughter cells. No chromosome duplication occurs between this phase and the beginning of the second division of meiosis because the chromosomes have already been replicated. Meiosis 2 is very similar to the steps of meiosis 1. The major difference is that the chromosomes did not duplicate between meiosis 1 and meiosis 2. In Prophase 2, the spindle apparatus forms again, and the chromosomes, in this case the sister chromatids, move towards the metaphase plate. In Metaphase 2, the sister chromatids are arranged at the metaphase plate. Because of crossing over events, the sister chromatids are not genetically identical at this step. As the sister chromatids line up at the metaphase plate, they are attached to the spindle apparatus made up of kinetochore microtubules that are attached to the poles that are defined by the centromeres. In Anaphase 2, the sister chromatids are separated and move towards opposite poles. Finally, in telophase and cytokinesis, the chromosome begins to decondense and the nucleus reforms. At the end of this round of meiosis, there are 4 daughter cells with a haploid set of genetically different chromosomes that are unduplicated. During meiosis, homologous pairs of chromosomes orient randomly during metaphase 1. This means, due to the random orientation, that the crossing over events are due to independent assortment. Each pair of chromosomes sorts maternal and paternal homologs in the parent cells, and these homologs are sorted independently of the other pairs. The number of combinations possible when chromosomes undergo independent assortment is 2 to the power of n, where n is the haploid number. So let's compare mitosis and meiosis. Mitosis conserves the number of chromosomes in the cell and produces cells that are genetically identical to the parent, and does so in a stage of division. Meiosis does not. Meiosis reduces the number of chromosomes from diploid to haploid and produces cells that are genetically unique from the parent, and does so in 2 divisions. The genetic differences that are observed in meiosis result from 3 events that are unique to meiosis only: crossing over, alignment of homologous pairs, and separation of the homologs. Another way to look at this is to study the table above. In the column on the left, there are traits of each type of division, mitosis or meiosis. Under the middle column, there is a description of how that trait is observed in mitosis, and in the right column, there is a description of how that trait is observed in meiosis. In preparation for the test, you should be able to recreate this table with fuller descriptions and drawings. Finally, let's briefly examine what happens when chromosomes don't segregate. There are events where chromosomes don't segregate properly during any of these divisions, mitosis, meiosis, or meiosis II. This can produce haploid cells in mitosis when it should have been a diploid cell, or it can result in a gamete that has too few or too many chromosomes. Two examples of this are trisomy 21, which results in an extra copy of chromosome 21. Another example is Klinefelter syndrome, in which there is an extra copy of the X chromosome, resulting in female characteristics observed in male individuals. Now let's use this video as a way to review what we've covered in this lecture.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis I is 2 haploid cells, Meiosis II begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In Prophase II, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In Prometaphase II, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase II. During anaphase II, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase II, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In Prophase I, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase I, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
So in today's lecture, we've covered meiosis. We've looked at the inheritance of genes, sexual life cycles, the phases of meiosis, mitosis versus meiosis, as well as what happens when chromosomes don't segregate. If you have any questions about this lecture or the additional information in your textbook, feel free to stop by and ask your professor during office hours. Bring your questions to class, or additionally, you can email them. You can also reach out to the learning assistant associated with this class. Now that you've finished the lecture, take the notes that you've just made and combine them with the notes from your textbook reading. After you've combined them, use the assisted reading questions as well as the questions at the end of the chapter and at the end of each chapter section as you prepare for the quiz and the test on this information.